These non-invasive tests, the IgE tests that we're talking about, offer a lot to the world. The blood test can be performed irrespective of the age or the skin condition or these things that are important with the skin tests. Multiple allergens can easily be tested. This is a classic example of a publication. This appeared in the journal Allergy back in the 80s, and basically what it says is that for a positive RAST result, that all of those positive RAST results would have been also positive for at least one of grass, house dust mite, and cat epithelium. If you think about it, there's over 600 allergens to test, and even components of those allergens that they test. You don't really need to test all of those in every situation, and a screen is very valid and can be very useful and helpful. Now, our simple screen that we produce is called the IVT Allergy 2, and it's a simple blood test that can simultaneously but separately look at these various allergens. It can be done in 2 simple steps. You simply add the patient and conjugate to the device itself, let it incubate for 2 hours. Kind of 2 minutes of hands-on time will lead you to the results, a substrate indicator. 2.5 hours later will give you the result. This procedure could easily be performed by anybody at almost any skill level, and then the result is interpreted by looking at a negative yellow, and it changes because of the bromocresol reaction with when the allergen is present, the specific IgE, from a purple to an intense purple. So it is somewhat semi-quantitative, but it does not express it in terms of IU per mil by any means. But it is a good, quick screen. This screen was intended to be read visually, but it can also be read with a scanner, and the results can be tracked and monitored on a computer. This is the one I want to talk to you about mainly today, and this is the IVT Allergy Nano. This employs some of the greatest technology that I've seen in allergy in the last 30 years. I think it is technology that will make allergy more prominent in our workloads, in our days, and in patient care especially. It will be technology. You know, the technology that we have with cell phones, computers, microwaves is now being applied in so many areas of medicine, and it's finally catching up to the practice of allergy. What it really does, it's comprised of a reader unit that takes a microtiter well, reads it, and then compiles the data, and we'll talk more about how that all happens, but it's a time-proven technology. ELISA technology has been around for many, many years. It's very successful, very reliable, very accurate and reproducible. Low-cost equipment. This equipment is relatively very low cost, and the software and reagents are available. Correlation with Phadia is quite outstanding. I've worked for Phadia. I've always used them as the gold standard in all of our 30 years of manufacturing allergy products. They are the best. This is flexible enough that you could run 1 patient or 96 patients on a single run. 2 hours, less than 2 hours of test result time. It can be run on multiple multiplex allergens, anywhere from 4 allergens per patient to up to 25 allergens. And it can be certainly qualitative, but it is quantitative. IUs per mil is very important. What happens is, in this test system, we take a microtiter well, and every spot that is printed on the bottom of a microtiter well exhibits a completely separate experiment or test in itself. Here we have some food, some molds, and some weeds tested even if at the bottom of a microtiter well. These, in the presence of the IgE from a specific IgE from a patient sample, will emit light as it is a chemiluminescent ELISA procedure. Here's what it looks like in technology terms. It's really state-of-the-art, this multiplex array. It's what really gives a lot of data very quickly and potentially at a very low cost. What happens here is the allergen is coated to the bottom of a microtiter well, and then if you have patient IgE present, it will bind with that allergen. A subsequent avidin-biodin reaction will take place, emitting chemiluminescence or photons as this chemistry proceeds in the presence of specific IgE from a patient sample. As I mentioned, it's a classical chemiluminescent ELISA technology. This is what the plate would look like to the camera, the CCD or the camera that's in the reader that I showed you. And here we would have the photons of light appearing, and this is a replicate sample here. If there is nothing from patient IgE or a negative sample, there will be no light emitted, of course. What we were able to see in working with allergy for the many years that we have was just an incredible signal-to-noise ratio. As we look at something that's positive, you might get 65,000 photons of light that are captured and counted and analyzed in this system on a positive result. On the other hand, a negative sample is virtually zero. The cutoff is really a very, very low number. So the signal-to-noise ratio tells us we have some really fantastic chemistry here, which is all important. It's flexible. These multiplex assays allow you to do things like coat 4 allergens on an individual microtiter plate at the bottom of the wells, or up to 25. In France, for example, they only have reimbursement for 9 allergens. And so 9 allergens, why would you coat any more? And that's the way multiplex gives you flexibility. Test procedure is really quite simple. It amounts to patient sample being added, a second conjugate, 2 conjugates being added as substrate indicator. This being done with 60-minute incubation, 30-minute, 20-minute, and then a 1-minute read with the substrate. So 1 hour and 51 minutes is the entire test procedure time. Very simple. This is very automatable, and we've chosen a Dynex instrument to work with on this to automate this very simple procedure. Data management and handling the data for this is critically important, and what we see here is the system that we have allows you to interpret that result, keep track of the microtiter well and the allergens that you're testing, and realize that you could have as many as 2,400 tests on a single microtiter well, 96 microtiter well. So that's a lot of data to manage, but look at the fantastic things we can do today with the IT and the software that we put together, the middleware that allows that data then to be sent out to an LIS system and give us tremendously big advantages that were never there 10 years ago. Basically, a microtiter well may look like this. The patients, we provide a known calibrator, a reference calibrator, with every run that you would do. If you had positive samples represented here by 1, 2, and 3, that's what you would get. And by a single point calibration, we're able to extrapolate and give a quantitative IU per mil measurement of the specific IgE from those individual allergens. We did a study at a local hospital, and I just want to show you a little bit of typical correlation data that you would see with Phadia. This is the local hospital, as I mentioned, who just last week provided us this data. It's not huge data, but I think you'll get an idea, and this is very representative. We tested foods, and with Phadia in the red, with our product in the blue, the pollens and halens representing various types of allergens that you'll see, and very good correlation here. These are on positive results. The foods that we looked at, if you look at the percentages and the correlation, we're about 94% on this food analysis by this particular hospital. Pollens, about 97% correlation to Phadia. Again, the inhalants, very high correlation. But anyway, what we're thinking is this multiplex technology and the new technology that will allow us to have high sensitivity, accuracy, and reproducibility that is absolutely required, but is quite easy to achieve with an ELISA technology. The customized allergen plates, if your physician or clinician is requiring, your lab is requiring 10 or 18 allergens, that's very possible. One patient can be run or 40 patients can be run. The microplate would itself be saved for another night to run another 28 on the second night. Sample size is very conservative at 10 microliters, less than 2 hours of time for the final results, and a very inexpensive price. This cost is pretty attractive for allergy testing. I know around the world it gets pretty expensive. It's very easily automated. We'll see that to be very promising. Low-cost equipment and software are available, and we can test, as I mentioned, up to 24 allergens per plate. What we're doing is working with the Merrill Diagnostic Group here in India, and we see a great opportunity for the India market. We collaborate with a lot of different organizations. The Centers for Disease Control in Atlanta, Georgia, that's where Dr. Modi is. We've worked with them for about 15 years on different projects. Thermo Fisher and Cardinal Health are a couple of the major distributors in the world and around the market. We private label products for them and many others. Our China distributor has literally provided millions of these allergy tests over the last few years, and Human Diagnostics and others across Europe, Fumos, Innova. LabCorp, we put our instrumentation and reagents into the LabCorp organization last year. Qantas is a local company we work with on a regular basis, and as I mentioned, Merrill Diagnostics here in India. So just to summarize, the collaboration that we have, we're planning on doing a little bit of manufacturing here. We will keep some of the key chemistry and work with that, but the opportunity to have local labor and to avoid some of the costs associated with tariffs and duties and shipping, will be able to take advantage as we manufacture part of this product here in India. It's a proven business model. We've done this in other places in the world, and it works very well. We see a very mutually beneficial opportunity here with this collaboration. Now, in India, I know I had the opportunity to visit some of your labs on Friday, just here locally, and I know that the sophistication is truly here. I saw some of the most incredible instrumentation I've ever seen in the world, and I know it would be the envy of the CDC or LabCorp or any of those with some of the instrumentation I saw just this week. I know that the desire is there. You have a population so great that any work that will be done in this allergy arena will be well worth the expense, the investment, the time that you put into it will benefit so many people. I know that I've met a number of you professionals, and I see such a